group video. So the question is, you're given a nucleotide sequence of RNA, of a eukaryotic messenger of RNA. And so the RNA polymerase has worked from right to left, and it starts at the 5' to 3' end. So the 5' end is shown here in the picture. Then the sequence of the template strand is just the copy, kind of the base pair of it, but instead of the uracil, like in the mRNA, it would be the T. Instead of U, it would be T. Next would be the coding strand, which would match up with it'd be almost the same exact strand as the mRNA, but again, U was replaced with T because of the difference of RNA and DNA. And then for D, it was to show a picture. And so you can see the coding strand is on the top, and the mRNA is in the middle, and then the template strand is on the bottom, because the mRNA will come in and bind to the template strand, and the coding will sit kind of above that. Question 2 was definitions. So TBP binds directly to the TATA box, while sigma factor binds directly to the -10 and -35 sequences. Guanylyl transferase is a eukaryotic enzyme that links the 5' carbon of one nucleotide to the 5' carbon of another nucleotide. Group 1 introns use the nucleoside guanosine as a cofactor during catalysis, while group 2 introns use adenine as a cofactor. The terminator is the endonuclease that assists in terminating transcription in eukaryotes. TFIIF is the GTF, which acts as a bridge subunit to link TFIID to TFIIF/RNA polymerase II. TFIIH is a GTF, which acts as a phosphorylate and activates RNA polymerase II. The spliceosome is composed of subunits called SNRNPs, and they're composed of two components, protein and small nuclear RNAs. Question 3 concerns the proteins involved in transcription and RNA modification, so it's just wanting to indicate which ones belong where. So 2 proteins that act as a helicase to separate 2 DNA strands would be RNA polymerase and TFIIH. A protein in prokaryotes that acts as the helicase to separate RNA from DNA during transcription would be Rho protein. 3 molecules in eukaryotes that function during RNA modification as ribozymes would be group 1 introns, guanosine, nucleoside, and small nuclear ribonucleoproteins, or SNRNPs. And a protein in prokaryotes that acts as a transcription factor would be the sigma factor.
Okay. Number 4 is our wonderful application problem here. I really like this kind of problem. It's really kind of cool when I figured it out. I am being kind of quiet because I am in the public library at the moment, and they say I can't talk. So we're doing genetics by homework, like in the library. Okay, so number 4 is saying that we hypothesize that a certain gene called our favorite gene contains 4 introns, and you would like to verify the presence of these introns using the electron microscopy method. So it says draw images of the resulting nucleic acid structures following the hybridization experiments described in the parts below. So A is saying the pre-mRNA for YFG is hybridized to the genomic DNA copy of YFG here. So I think what I did was I accidentally wrote out a couple of steps that I didn't need to, and then a couple of things I wasn't asking. So, but essentially, here we're taking the DNA, the template strand, and transcribing a pre-mRNA oh my goodness, pre-mRNA strand from here. So B is the one step further than that is you're transcribing. You get your pre-mRNA here. You go through a splicing method, so it removes the introns off of this pre-mRNA strand, and this mature RNA strand binds to the template strand here. Now, because we spliced out the intron sections of the mRNA here, you're going to find these intron DNA loops of the template strand. They're going to make these little bunny ears. And we've got 4 of them spliced out, so you'll form 4 of these. Notice that the mRNA strand is going from 5' to 3', binding with the template strand, which is 3' to 5'. The next step, the DNA coding strand overlays on top of that, and you get these little R loops in the middle with all my arrows there, and they're binding over the top just like that, kind of making those little bunny ear things right there, just like this. So question C is asking, well, the mature RNA for YGF is hybridized to the genomic DNA copy. Okay, so that means they're overlaying on each other. So it's saying that the 5' intron has a defective donor site and branch site adenine. So we just know that as it's the same as this drawing here, except for the 5' most intron loop, it's missing because it couldn't happen, because it couldn't bind. Now it's essentially the same deal with letter D here, except it's saying the 2nd and 4th introns, and the pre-mRNA both contain defective donor sites and branch sites, adenine. So it's the same as B here, but the 2nd and 4th ones will be gone as well. So you only have 2 loops. For number 5, it's giving us a list of proteins and nucleic acid sequences involved in transcription and RNA modification. So letter A is asking which of the above is found in E. coli. And we know that the 35 sequence and NUSA are found in E. coli. So letter C is talking about which of the above is involved in transcription initiation. So 25 sequence, the enhancer, TFIID, the SNRPs, silencers are found in transcription initiation. Letter D is talking about which of the above is involved in the transcription termination. That's RUT, NUSA, the SNRPs, and the 25 sequence. Letter E is asking which of the above is a DNA sequence, and GC box 25 sequence and 35 sequence is part of the DNA sequence as well. And I think that is it.
Awesome.
Sweet deal.